So, yes, bless you. Hypothesis involving manipulation, by definition, you manipulate it so you can control whatever you want to control. That's what manipulation means, right? So you do whatever it is that you're going to do to the animal or plant, and therefore you control everything that happens to that animal or plant. When you're talking about a hypothesis of choice, you have less control over it. So which kind of food does this woodpecker prefer? Whatever the issue is, it's a little more complicated, a little more, because there's factors you can't control. What did that woodpecker grow up eating? Where is that woodpecker? What's in the environment? Is it cold or is it hot? You can't control those things. You have less control when you're looking at choice. The last and the one you have least control over is when you're just observing. When you have a hypothesis where you're going to observe what's going on, you have no control over what happens. It could be overnight. You could get a winter storm coming, right? You could get a drought. You just don't know what it could be. A deluge, right? You get absolute flooding in the area, and you just don't know what's happening or what's going to happen. So you have the least control here and the most control here over all the different environments. So I think that should help you answer these questions. Of course, these three questions have to do with which one of these three, right, would you choose for those particular questions. Are we good? Yes, ma'am. That's when you're looking to see what, and you can read it. It says when used when investigating species preference. So what kind of habitat does this species of woodpecker like? Does that make sense? So it's just choice. What is it that this animal is going to choose? To go right or left? To pick mealworms or grubs? It's all up to the woodpecker. But the problem is, there are factors that we don't control. Do you understand that? Can you understand that portion of the concept? Yes? I can't hear you. The person, I don't even know who had. Remember, I got a light in my face, so I can't see you. Who asked the question? So did you understand that? Okay, very good. That's right. You did another test. So when you accept a hypothesis, and let me be very clear. We're talking about accepting a hypothesis or rejecting the hypothesis. So when you're rejecting the hypothesis, you're saying that you have this experimental group and you have your control group and you have your some unknown group, and you compare them, right? And either, yes, the evidence supports the hypothesis, so you accept it, or no, you reject it because it does not support your hypothesis. So you do an experiment today, and you find there is metal in there. I accept that it's a clothespin for today. Then tomorrow somebody else does a different experiment. Maybe they use a stronger magnet. Maybe they test it with some other method that you didn't think about. They do an MRI, they do an X-ray, whatever. And they find strong evidence that suggests it's not a clothespin. So what are you going to do? You're going to reject it. So you accepted it yesterday, but today you're going to reject it because you found new evidence. Anytime you find new evidence, it changes. That is science. That's the strength. Cleveland School of Science and Medicine, Mr. Mendoza speaking. May I help you? To go home or to come back? Ariel Johnson to the office, please. You're coming back. Tell Ariel Johnson she's to go to the office. Somebody tell her. All right, are we clear on this? Is everybody okay with this? Okay, so that's it. That's really all there is to it. Let's go on to the next. All right, so here you have hypothesis and predictions. You have to read. I know that a lot of you don't like to read, or you try to answer the questions without reading for whatever reason. Say that again.
says it right there, null hypothesis is in the description.
Yeah. It tells you what the null hypothesis is, but it's not an easy thing to understand. That's why I wanted you to do the bag lab. Now, I told you that, we're going to go ahead and assume that you have this paper bag, right, with this tape around it, and there's a number on it, and inside you have something you don't know what it is, right? That's where we're at. And a lot of you said, I know there's a clothespin in there. And that's what some people said. I know there's a clothespin in there. There's absolutely no doubt in my mind that there's a clothespin in there. And this is the metal spring. Isn't that what people said?
Yeah.
That's what I heard people say. And so that was a hypothesis that a lot of people said. In fact, that's the example I used, isn't it? Yeah. In the homework that I gave you. So now here you have this clothespin. You're trying to figure out what it is. After all the data is done, how many tests do you have to run? How many tests do you have to run? How many different things do you have to do to prove that it's a clothespin? Let's assume that we proved there's a piece of metal in there. That doesn't prove that it's a clothespin, does it? Let's assume that we do. How many tests do we have to do to prove that with 100% certainty that what's in that bag is a clothespin? Of course, without opening the bag. You can't. It's not possible to prove it. Do you see that? Can you understand that there is absolutely no way for us to prove anything in science? Somebody will inevitably say, we've proved something, or can you prove? It is literally impossible, and I say that, and I'm going to tell you there's exceptions, right? But it's literally impossible to prove anything in science. The only thing, now let me ask you a different question. No proof. Let me ask you a different question. How many tests does it take? Let's say if I said there's no metal in that bag. What if my tests prove that or show that there's no metal in the bag? All the tests are negative. How many times do I have to get a negative test to disprove that there's a clothespin in that bag? Yes? Yeah, I'd like you to answer it. How many tests do I have how many tests do I have to run that would disprove that there's, in other words, they would say there is no clothespin in that bag? Yeah.
How many ever is it?
Okay, let me phrase this in another way. I run my test, my metal test. I'd say I have a logical argument that says that there has to be metal in the bag for there to be a clothespin, right? We said that. Right. If there's no metal in the bag, then there is no clothespin. Do you agree? If I do my test and I with all my control groups and I show clearly there's no metal in the bag, is there a clothespin in the bag? No, there isn't. If I agree that there has to be metal in the bag for there to be a clothespin, and my tests show definitively there is no metal in the bag, then there is no clothespin. Do you agree? Isn't that logic? We call that logic. So what we're saying is, remember the if then, if there is no metal in the bag, then there is no clothespin. The if-then statement. That's why the if-then statement's a great hypothesis, because it's testable. So I tested it. There's no metal in the bag. Is there a clothespin? No. So how many tests do I have to run? Once. I only need to do it, I'll have to repeat it to make sure. But the bottom line is, you only have to disprove something, you only have to do it. You only have to find the negative once. Let me give you another example, an easier example. Are you ready? If I throw an apple up in the air, what's going to happen? It's going to fall. It's going to come back down. So there's a rule that we came up with, isn't it? For everything goes up, everything that goes up must come down. Now, that's a law, right? It's called the law of gravity, right? So let me ask you something. How many times do I have to throw it up to prove that every time I throw it up, it's going to come down? How many times do I have to throw it up?
Once.
Is that all I have to do to prove it? Well, how many times is it as many times you need to?
Every time you throw it up, it's going to fall right back down.
There is no number. It's infinity. I have to do it an infinite number of times. I have to throw it up to prove that every time, for all time, every time I throw it up, it's going to come down. I have to do it out for it. I could never stop doing it. The minute I stop doing it, there's going to be a question. Somebody's going to ask a question, and I bet you'd be one of you. How do you know the next time it won't keep going? If I throw it up 1,000,000 times, I could always ask the question, right? Even if I do it 1,000,000 times, we're old people, we're like Asgardians, we've lived thousands of years. They had one poor guy that has to throw the ball up and watch it come down. Every time somebody could say what? Next time it's going to keep going up. Couldn't they? Now here's a question. Ready to ask the opposite question? Are you ready? How many times do I have to throw the ball up and it not come down to disprove the idea that the ball goes up, everything goes up, comes down? Once. It only has to not come down once for us to disprove it. So it turns out that in science, what we like to do is do what's called the alternative, and I would take notes on this, hypothesis. We like to try to disprove something because we can't prove it, but guess what we can do? We can disprove things. Because it's easier. We can't do things for infinite a number of times. So we say, oh, well, any test you do, you can write a null, and this is, by the way, this alternative hypothesis is known as what?
Null hypothesis.
The null hypothesis. And that really is all there is to it. It's the opposite of, it's the negative of whatever that hypothesis is. So now let's take a look at this. It says, study an example listed above, study the example above, illustrating the feature of cryptic and conspicuous caterpillars. Then answer the following. Generate a hypothesis to explain the observation that some caterpillars are highly colored and conspicuous, while others are cryptic and blend into their environment. So, did anybody come up with a hypothesis? So who said they didn't understand the question? It's okay that you don't understand. I just need to know why you don't understand. What don't you understand about that question? Did you read the paragraphs up here? Did you read the observation? So the first set of the instructions say to study the example above. So you have to study this example. Here are the observations, here's hypothesis, predictions, here's the assumptions, here's what the caterpillars look like. Do you agree? Observation 1, observation 2, there's some assumptions here. What's going on? Do you understand all that? Did you understand? You guys got to answer me. Did you understand that part?
Yeah.
Yeah, okay. So you read it. Is it? Am I? I'm not going to read this to you. Have you read it?
Yeah.
Okay. What part of this did you not understand? Generate a hypothesis to explain the observation that some caterpillars are brightly colored and conspicuous. That's what conspicuous means, right? Brightly colored means conspicuous. So some caterpillars are brightly colored, while other caterpillars are what? Cryptic, which means they what? They blend into their environment. So who has a hypothesis as to why? Yes.
Okay. I said if the insectivorous birds pay more attention to brightly colored caterpillar species than the caterpillar species that is more cryptic. Then insectivorous birds see color or are more fascinated with the brightly colored species. And then I was using like an idea with what the assumption said.
So I'm going to rephrase what you said, I think, okay? So I'm pretty sure I got this right, but if I'm wrong, I apologize. But I think what you're saying is, I'm going to rephrase it. If the we're gonna go ahead and assume these things, right, are true. If the what birds are the ones that chase them? Did they mention it? No. Yeah. Birds, which are insectivorous, are present, then the what are they called? Conspicuous caterpillars will be chosen more often than the cryptic ones. I don't believe people are putting things away. Do you all agree with that?
Yeah.
So that's an if-then statement, and that explains all that. Now state the null hypothesis. What would be the null hypothesis? So you got to figure that out because we've run out of time. But the null, and you see why I wanted to hurry up with what we were doing. So that, the alternate hypothesis would be that if the birds present, if the birds are present, then the conspicuous caterpillars will not be chosen. Right? Do you agree? If this is the hypothesis, then the null hypothesis would be this part is they will not be chosen. Does that make sense? All right. If you need more help, come in, stay and get a pass from me for advisory before you leave. All right, have a good one. Okay, so in this portion of the scientific method discussion, it asks, during the course of these investigations, and actually investigations in general, sometimes new information arises that results in the observations that are unrelated to the experiment you had first thought of running. Now, investigation is another word for experiment. Now, they give you three scenarios, and they tell you that the observation described in each case was not related to the hypothesis the experiment was designed to test. So, in each case, there was an observation that really didn't have anything to do with the experiment it might have been related, but didn't have anything directly to do with the experiment that they were running. So here's an observation. During an experiment on bacterial growth, these girls noticed that the cultures grew at different rates when the dishes were left overnight in different parts of the laboratory. So, what could you say about that? Well, one possible thing you could say is that for a prediction, and what is a prediction but a hypothesis? Now think about what a hypothesis is. A hypothesis is a prediction, right? If this is true, then this would happen. If this is true, then that would happen. So one possible form of a hypothesis would be a prediction. So if the bacteria were left in the brighter part of the laboratory, then the bacteria grew at a faster rate. Now that's just an if-then statement that has to do with that observation that these ladies had while doing some other non-directly related experiment. So you start off with a hypothesis, just like any good scientist, And then you have to design an investigation. Now remember, an investigation is another word for an experiment. So what are the parts of the investigation that you've learned already? Well, if you have to design an experiment, the first part of the experiment you're going to design is the experimental variables. So let's look at the experimental, the ones that you're going to change. What are you going to change? If you think it's light, you think the brighter the light, the faster it grows. It might not be. Maybe you thought it was dark, or maybe you thought it was the medium one, whatever it is. So if you think light had something to do with it, and why did I think that light had something to do with it? Because of the observation. The observation stated that during the experiment, these girls noticed that cultures grew at different rates when the dishes were left overnight in different parts of the laboratory. Now, there's an assumption here, and the assumption is that it was light. There was some light in the lab, right? And that the light had, and of course you might think, well, maybe it was black in the lab. Maybe it had nothing to do with light. Maybe it had to do with temperature, or maybe it had to do with moisture, or maybe it had to do with some other factor that we are not considering. Well, it could be, but it could be that there was a light on in the lab. Maybe in different parts of the lab somebody left lights on, and in the other parts they didn't have any lights on. So in any case, in this hypothesis, the group chose light. So the idea would be place the bacteria in different lighting conditions and see how fast they grow. So you'd have one group of bacteria growing in 10% light, another in 20% light, another in 30% light, another in 40% light intensity. And you see how fast they grow. Well, that's great, but what do we have to do if we're going to have any experiment? Well, you have to control all the variables. So you have to control all the other variables. You have to control the temperature, the type of light, the food, or the type of bacteria. All the different strains of bacteria have to be the same. The temperature in all the different areas have to be the same. The food have to be the same. The food that you're providing for the bacteria to grow has to be the same. The initial bacteria numbers have to be the same. So the only thing you're only going to change one thing. And what's that one thing? Light. So that's number one, the hypothesis. Number two, what is the thing you're going to change? You're going to change light. And the other part of that is you're going to control, part two of this investigation, you're going to control all the other variables. And lastly, you have to have a control group. We have two control groups, the positive and the negative. 100% light, that's your positive. And 0% light or dark, you're going to have is your negative control. Then you just collect your data, you analyze it, you come to a conclusion. So that would be the first observation. So you should go ahead and do the second and the third on your own and turn it in as soon as you can, or on Monday. One thing you should know before you try to do the second half of this page is, what is agar? Agar is usually used in a petri dish to help grow bacteria. And the reason we use agar is it's a great medium. It's pretty sturdy. And when you create agar, you usually make it in some kind of Erlenmeyer flask, a fairly large one, and you can pour many different plates. So you can pour numerous plates with one sample of agar. And the plates are also known as petri dishes. So in these petri dishes, you can pour the agar. Agar is seaweed protein. Someone mentioned in class that some people use it in food preparation. They saw it on a cooking show. But agar is a protein isolated from seaweed protein. Is a protein isolated from seaweed. And it turns out that when mixed with water in the microwave and then allowed to cool, it forms a plate. It forms a gelatin, a pretty stiff gelatin, stiffer than Jell-O, but it's a lot like Jell-O gelatin in many ways. So it forms this matrix and acts as a really good growing medium for bacteria. So it stiffens up, and of course you can make it stiffer or less stiff depending on how the concentration of the protein. But you pour it in here, just like you would Jello gelatin. You pour the agar into the mixture. But what's interesting is you can add anything else you want to that mixture. You can add blood, for instance. Some bacteria require blood to grow. You can add blood, or you can, so that you add the food that the bacteria eats. So you can control the concentration of the food, right? You can add antibiotics if you want to see if a bacteria is resistant to antibiotic or not. You can add antibiotics and then run a test to see if it's resistant to antibiotics. Or you can add hormones. And in the case of the second half of this page, the scientists added hormones to the agar. And then they allowed the plant clones to grow and sink their roots into the agar. Okay? And so what your job then is to think about what experiment, hypothesis, and experiment you would design as you try to solve this problem. Now I'm going to tell you that, again, the agar, you can add different concentrations. You can add whatever you want. So this could be, 20% agar hormones, or this could be 50% hormone, just by adding more or less hormones to the mixture. So remember that as you're looking at how to design that experiment. On this page, you're looking at the difference between precision and accuracy. And if you look at these targets, it's pretty straightforward. You have, in this case, it's a target, so imagine someone shooting in a target range or BB gun or what have you, or arrows or whatever it is you're looking at when you're looking at a target. Someone who's a really good shot could shoot the bullseye often. But if you look at this, if you have this is a shot group that's very inaccurate. In other words, it's not near the center, not near the bullseye. But all their different trials were very close together. Even though they're not on target, they're repeatedly close together. They're equidistant from the center of the target. Where here, this grouping is less precise. In other words, they're not as close to one another, but they are closer to the center of the target. So that's the difference between accuracy and precision. Now, this grouping is both imprecise and inaccurate. So they're not near the bullseye, and they're spread apart. Where this grouping is both precise, in other words, they're all huddled together, and they're accurate. They're near the center. So in this example, they used arrows in a bow and arrow competition, and that's what you're looking at. So whenever you're measuring, and whether you're using a caliper, the way you see here, a micrometer rather, as you see here, or you're using Erlenmeyer flask to measure volume, or you're using a ruler to measure length, whatever it is you're doing, there is a value that is accurate, so near the actual value, right? And the other thing that you want to do is you want to make sure that every time you measure it the same. So those two things are important. So even if you're not accurate, as long as you're measuring it all the same way, at the very least you have something that you can correct whatever your error is and bring it back to an accurate measurement. All right. Why do you have to be both accurate and precise? I think you can answer that. Why do you want to be both accurate? Why would you not just want to be accurate? You're close to the bullseye. That's good enough, isn't it? Think about why you would want to also be precise. I think that being precise is very good. If you're always getting the same measurement, that's great. But what if you're not on the... Why is it important to be accurate or be close to the actual value? Now here it says a researcher is trying to determine what temperature enzyme A becomes denatured. Now you don't know what denature is yet. And you don't know what temperature is, but you don't know what an enzyme is yet. We haven't discussed it. And you don't know what denaturing is. I understand that. However, I think you can still answer the question. You don't know what the enzyme is, what an enzyme is really, and you don't know what denaturing is. Okay. Let's see if we can answer the question anyways. Their temperature probe, I think you know what a temperature probe is. A temperature probe is just a device that can measure temperature, usually connected to some kind of electronic recorder. But it could be a thermometer, but they would probably use thermometer if it was a thermometer, not probe. Probes are usually connected to some kind of electronic measuring or collecting device. Is incorrectly calibrated. Now, see, here's the question. What does calibration mean? What does it mean to have something that needs to be calibrated? And why is it important to calibrate your device? All right. So, the best example I can give you as far as an example that you might be used to is a scale, a bathroom scale. You probably have one at home, or you've seen one where you stand on it, and it measures. It goes, from 0 to whatever. Usually it's 300 pounds on the other end. So, on these devices devices have to be calibrated. And what that means is that for whatever reason, as the device is used over and over again, the springs start to measure inaccurate weights. Usually the same inaccuracy. So let's say it keeps measuring you 20 pounds heavier than you really are. But every time you measure yourself, you're still 20 pounds heavier. Most scales have a little screw that you can twist to recalibrate it. So what you do, and usually if that's true, if it's always measuring 20 pounds heavier, the arrow, instead of pointing, points to 0 when it's actually 20 pounds on the scale. So what you can do is that you can put a standard weight on it, put a 20-pound weight on the balance. You know it's 20 pounds because it's made to be 20 pounds. It's been pre-measured and standardized. You put it on the balance. The arrow's pointing to 0. So you move. The arrow would, once you put the 20 pounds on the balance, the arrow would point to 40, right? After you put the 20 pounds on, instead of 20. So you twist the screw until the arrow points to 20. Then when you take it off, the arrow goes back to 0 because it can't go any less than 0. So now you've calibrated the scale. So what you used a known weight to reset the springs on the balance. Well, a lot of machines need to be calibrated. You might have a parent or a grandparent that's diabetic, or a cousin. They have a blood glucose meter. They use a little strip. It has a little bit of a window, and they stick it in a machine, and the machine reads to them some kind of number. Usually it's in the 90s or 100s or whatever. The units are not important for this discussion. So they stick it in there, and it reads 90 or 100. These devices have to be calibrated, and what that means is that over time, these become inaccurate, just like the scale became inaccurate. Even though it might measure the same, in the case of the scale, and let's go ahead and use the bullseye example. in the case of a scale, instead of measuring 20 pounds as 20 pounds, it was measuring 20 pounds as 40. So it was here. It kept hitting around here because it's 20 pounds off. The spring needed to be calibrated. Once you calibrated it, it moved your measurements closer to the actual value, closer to the bullseye. Now with a glucose meter, if you keep reading 90 repeatedly before and after dinner, let's say you read 90. If it's not calibrated, you could be higher or lower than 90 because you're not being accurate. So you're never sure with any machine whether you're getting accurate measurements unless you calibrate it. So in the case of this glucose meter, you might be hitting 90, and every time you measure it, it's 90. Every time you measure it before dinner, it's 90, and you might be getting measurements that are here, very precise, but not accurate. So there's a liquid that you can that comes with the glucometer, the glucometer, that you can use to dip the little on one of these test strips into, and stick it into the machine and hit calibrate. And this is a known value. Let's say it's 100, and the machine knows that this is 100. So then the machine automatically reads this and resets its internal mechanism so that it's measuring more accurately. And so now, after you've done this, you go ahead and test your blood, and now your values are closer to accurate. Now it's reading 110 instead of 90 every time before dinner, because that's your actual blood glucose. Hopefully that's not true, but anyways, so that's an example. Another example is what we do in a laboratory all the time, which is measure masses, and we often use an electronic scale. And the electronic scale, again, just like the electronic balance, very much like the electronic scale that your mom or dad or somebody has at home. Has a round plate usually. You put a little weighing paper on it, and you weigh it, and it gives you a reading, usually in grams. Let's say it's 2 grams. Well, anytime we use an electronic balance, the very first thing you do is you're going to hit a button, and depending on the model, it's going to be different. You're going to hit a button that says TAR. And what TAR is basically a calibration button. The button is just going to zero it out. You put whatever it is on the let's say the weighing paper. Let's say you have this little cup that has your mass, and you want to make sure that you measure the stuff that you're putting in the cup, whatever it is, the powder or the liquid. You want to measure the mass of this stuff. You don't want to measure the mass of the cup. So you put the weight of the cup, you put the cup on the balance without anything in it, and then you hit TAR, and that makes that 0 grams. Now, of course, the cup has some mass, but because you push TAR, it zeroed it out. So whatever is going on with the machine, anything you put in it other than the cup is going to be the mass of whatever's in the cup. So when you add the stuff in it, and it says 10 grams, then you know those 10 grams is the mass of the stuff that was in the cup, because you've already accounted for the mass of the cup by pushing TAR. So those are kind of 3 examples of calibrating machines. The whole point of calibration is that you have to account for wear and tear of a measuring device. You're accounting for inaccuracies of the machine. So if you have a temperature probe that is incorrectly calibrated, how might this affect accuracy and precision of the data collected? Well, there's I'll let you then think about how to answer that. Accuracy and precision. Knowing that you have this probe. Usually they look like this. In fact, we'll be using one. And some of them have different ways of collecting data, but they'll have a little window or they'll have a wire that connects to your computer in some way. And then this, of course, measures the temperature, and this records the temperature in degrees Fahrenheit or degrees Celsius. And if you connect it to a computer, then it collects a bunch of data very quickly of temperature. Now, if this is calibrated incorrectly, it might be reading 10 when the temperature is actually 20. A good example is those of us who are in class that looked at the thermometers and it kept reading 60 degrees Fahrenheit, and all of us were saying there's no way it's 60 degrees in here. So anyways, that is a calibration error, at least in this example, that this thermometer is miscalibrated. Instead of 10 degrees, maybe it should be 20 degrees Fahrenheit. So there's something wrong with this machine. And because of that calibration error, it's always going to be 10 degrees off. Now, this thermometer is always going to be 10 degrees off, but there's no guarantee that every time you run the thermometer, every time you run the experiment, somebody's going to use this probe. It might use another probe, or a third probe, or any one of the probes that they pull out of the drawer. So that's one way you can affect precision versus accuracy. Of course, by definition, this is inaccurate. All right, I'll let you then finish that description. I think this page is self-explanatory, but I'll quickly say that qualitative is any data in which you have to define something that has really very little to do with numbers. It has something to do with the characteristics of the thing that you're looking at. They're descriptive. So all the red ones. Are they red or are they green? Are they small or large? So these values, small, large, red or green, dead or alive, these are not numbered. They're qualities. Now, when we're talking about ranking is a comparative number, so a comparative thing. So you're looking at something that has the order. Am I the youngest or the oldest? Am I first, second, third, or fourth? Is it medium or dark or darkest or light? Is it 1, 2, or 3? These are, in other words, there's gradations. There's order to your system. It doesn't have to be a number necessarily, but it has to be from high to low, for instance, or from big to small, to smallest. So anytime that you're ranking them, it's a type of variable that we call ranking. Of course, quantitative is kind of easy, and that has to do with numbers. So you're talking about weight or height or mass or whatever. In the case of discontinuous, it's the number of children in a family. It could be 3 or 0 or 4. There's no way of predicting a value because their numbers are not continuous. Where if you're talking about the height, the numbers are going to be continuous, right? So you're going to go from, you might have somebody who's 5 foot, someone else in your family is 6 foot, someone else who's 5'8", and so you can put them in order because they're continuous. Where discontinuous, is a number. It is still a number, but they're numbers that aren't. You're not necessarily going to get 1, then 2, then 3, then 4. All right. So for each of these, I think you can easily decide if they're quantitative, ranked, or qualitative. Why is it desirable to collect quantitative data where possible in biological studies? Gosh, think about it. Why would you want to have numbers? What can you do with numbers that you can't do with anything else? With either one of these two. You can do some of it with ranked data, but you can't really do much. You can't do as much with numbers as you can with qualitative data for sure. You might measure in problem 3 or question 3 here, it says how you might measure the color of light, red, blue, or green, qualitative. How could you measure that in a quantitative way, red, blue, or green? Some of you might know that, so I'm not going to answer that just yet. I'll wait till our discussion in class. Give an example of data that could not be collected in a quantitative manner, explaining your answer. Some data that you can't use quantitative, you can't put numbers to it. So you think about it, and I think you can answer that. Sometimes ranked data are given numerical values. Rare is 1, occasional 2, frequent 3, and common 4, abundant 5. Suggest why these data are sometimes called semi-quantitative. Semi meaning halfway quantitative. Why isn't it completely quantitative, and why isn't it completely qualitative? I think that's also an obvious answer, so I'm going to let you try to answer that. So here you're dealing with manipulating raw data, and so you got data. In this case, let's take a look at it. You have a tally chart, so you're kind of counting how many you have. Here's how many people that are how many things are within this height range. You can count them. There's 3, and how many that are between 1 and 1.99. You put that's 4 here, right? And etc. all the way down, or actually 6, right? So 3, 6, 10, 12, 3, 2. And this is how many are for each of these heights in centimeters. I guess the height of seedlings. You're measuring this height in what looks to be centimeters. Hard to read it here. And of course you can do percentages, which is pretty straightforward. You take the math. Here's an athlete. You take its mass in kilograms, and you divide the lean body mass by the total, and you get the percent lean body mass. So you take whatever it is and divide it by the total, and that gives you your percentage, right? So that's how a percentage works. So if you have 2 right, and there are 100 questions, you got 2 out of 100. Well, 2 out of 100 is 2%, right? Okay. And of course you have rates. So you have minutes here from 0 to 4 to 60, it looks like, and you have cumulative. You have sweat loss in milliliters. You have 0, and then 340. And rate of sweat loss here, and if this is every 2 minutes, in 2 minutes it's 0, and here at 10 minutes you get 50, so that's 50 divided by 10, so that's 5, and after 10 minutes you're at 5 milliliters per minute, right, because you took 50 milliliters and you divided by 10 minutes, or time in minutes, so that's 10 minutes. And you keep doing that because that's what rate is. Rate is whatever number, in this case loss in milliliters, divided by time in minutes in this case. So here you get how many milliliters per minute. By the way, the notation here is ml minute -1. Whenever you have an exponent that's -1, you have to take it and invert it and put it over 1 over that. So it would be 1 over a milliliter. So if you have total rate of loss of sweat loss here over milliliters, over minutes, it would be milliliter over minute. Because here you had milliliters, and this is minutes, 1 over minute. It may not make sense, but let me just go ahead and show you mathematically how that works. I'm not sure, it's almost not worth explaining it to you since you may not have covered it in math, but if you have milliliters minutes to -1, this factor right here, this part of this equation means 1/minute. What does it mean when you multiply milliliters over 1/minute? Of course, you're going to put that over 1. When you multiply milliliters times 1, that's milliliters. 1 times minute, that's minute. It's milliliters over minute. So this notation is the same as milliliters over minute. So when you see it here, milliliters minute -1, it's the same as milliliters over minute, or milliliters per. This means also another way of saying this is per, milliliters per minute. So here it's 0 milliliters per minute, here's 5 milliliters per minute, here's 8 milliliters per minute, here's 9 milliliters per minute, and 11.3 milliliters per minute. I hope that made sense. Okay, and we can, if you have problems with that, we can discuss it later. Ultimately, it's not incredibly important you get it, but it's good that you're familiar with that kind of notation. All right, so here's some rates, and here's percentages, and there's tally charts, kind of 3-way of manipulating raw data. Explain what it means to transform data. Well, transformation of data, and if you look here, I believe it states it there for you, in a form that makes it easier to identify important features of data, trends, for instance. Some basic calculations such as totals, sum of all data. So you can take this data and you can find averages, for instance. This is raw data. This is the number. This number you measured, right? But you can find the percent lean body mass by manipulating it or transforming it. This is the raw data, but you can find the average height, couldn't you? So if you have take each of these and divide it by the total, you can find the average. You can find an average, right? The rates themselves are a manipulation of the raw. This is the raw data, and this is the manipulation of that data. All right. So anytime you're measuring or counting, this is what was measured. This was measured time. This was measured in the volume. This was measured. This was measured. This was measured, and this was measured. But and this is also the tally was measured. So the total was adding it. So this is actually transformation here when you summed them up. When you summed up the numbers, calculating percentage is a transformation of the raw data. Calculating the rate or dividing the milliliters by minutes is also a manipulation of the raw data, etc. But you can further manipulate it, couldn't you? So you can plot, for instance, you can plot the rate on a chart, and you can look at the slope of the line. The slope is a manipulation or transformation of the data, right? So the slope of the line tells you the rate. So if you plot these points, it's the slope of the line that's milliliters per minute, because X is in minutes and milliliters. Oh no, this is an acceleration. So you're looking at the rate of change of the rate. So that's a little more complicated. But in any case, the slope is a rate of change. There's all kinds of ways you can transform the data. That's what it means to transform the data. Why do we transform it? To find meaning in these numbers. The numbers don't mean anything by themselves. So an athlete has 76% body mass. Well, what does that mean? The only way that that means anything is if you find the average, the median, the mode, and that gives you more meaning to that number. If somebody says 76, that doesn't mean anything to me. But if you tell me 76 lean body mass, 76% lean body mass, that tells me what? Well, if an athlete's 76, but an obese person is 54, then that gives me some idea of the difference between someone who's obese and someone who's athletic, and it shows me how far I have to go to get where I need to be if I'm obese, or if I lose 1%, if I'm 1% less lean body mass next month than I was this month. Well, how bad is that a negative change for me? Sort of like grades, going from an 80 to a 75. That doesn't mean anything unless you know that what an 80 means, that an 80 is close to B and a 75 is close to a D. And so knowing that, then you know where that you're going down, and that you need to you need to improve drastically in order to go in the direction you want to go. So meaning is transforming data helps us get meaning from it. For each of the following examples, state a suitable transformation together with a reason for your choice. Determining the relative abundance from counts of 4 species of 2 different habitat areas. So relative abundance from counts of 4 plant species in 2 different habitat areas. All right, so think about it. What is that? What could that possibly mean? Well, here you have a habitat. Let's say it's mountains. Say here you have the mountains. And here you have a prairie. So it's a grassland. In the mountains, before you get to the the icier areas, perhaps you have kind of more forest because there's more water. Let's say that. And you have 4 different plant species. So here you might have a couple trees. So that's 2, and maybe here you have some grass, but you also have a lot of flowers, and you have here you have some flowers. So there's 4. Maybe here you have a relative abundance. Here you have 5 types of 5 trees and 2 short grass and 100 tall grass and 20 flowers. Here you might have 20 trees, 10 short grass, 20 tall grass, and 5 flowers. That's let's say per square meter. No, that doesn't make any sense. Per 50 meters. Every 50 meters you're going to get something that looks like this. Some square you draw a little section, and that's what you're going to find in each one of those 50-meter sections. All right. Determining relative abundance from counts of 4 plant species with 2 different habitat areas. Suitable transformation. What would you do? How would you transform this data to make it make sense? Well, you made a count, so you can take and divide. If you have 5 trees per 50 meters, then I could simplify that and say it's 1 tree per meter. 1 tree per meter. And I could, this one, I could say 20 divided by 50. And And where here I'm getting 1 tree per meter, over here I'm getting 0.4 trees per meter. Where here, over here I'm getting 2 divided by 5, or by 50, 2/50ths, right, of short grass. Over here I'm getting 1 or 10/50ths of short grass, or 1/5 short grass, 1 out of 5 short grass, 1 short grass per 5 meters, or you can say, well, it's 0.2 trees per meter in the mountains, and it's 0.04 trees per meter, or not trees, I'm sorry, 0.2 short grass per meter in the mountains, and it's 0.04 short grass per meter in the grassland. So, how is that? Why is this a suitable transformation of the data if I'm doing counting? Well, by finding averages when I'm counting, it lets me compare more accurately one type of environment to another. In other words, I have this set of data. This is just to count. But if I find averages, if I standardize it by dividing by the same number of, like in this case, area, then I'm able to compare the two environments more accurately. So then lastly, it's determining the effect of temperature on the production of carbon dioxide by respiring seeds. What's a suitable transformation? Well, let's take a look at it. How would I measure this? How would I measure the carbon dioxide being produced at a certain temperature? Well, I'd probably set the temperature at 20 degrees Celsius, and then I would allow the seed to germinate, to start to use oxygen, and there's stored sugar and produce CO2. So as I measure this over time, I'd have time, and I would have CO2 production, and probably in milliliters. And as time goes on, maybe it's 1 minute, maybe this is in minutes, maybe I produce in one minute, I produce 5 milliliters. I'm making this up, but in 2 minutes, I produce 10 milliliters. In 3 minutes, I produce 20 milliliters. Okay? Then I do another experiment. I do another trial where I'm doing 30 degrees Celsius time in minutes, and I do CO2 in milliliters. And let's say 1 minute, and it's 10 milliliters. And 2 minutes, it's 20 milliliters. In 3 minutes, it's 40 milliliters, right? Well, what would I do then to look at, if I'm going to do the effect of temperature on carbon dioxide? I would probably want to compare rates, right? I'd probably want to compare rates. I'd probably want to see at 20 degrees how much CO2 per minute am I making? How much CO2 per minute am I making when I am at 20 degrees Celsius? And so here obviously I'm doing 5 milliliters per minute, but here I'm also doing 5 milliliters per minute. Over here, here's a 10 divided by 2 is 5. Here, you know what? I should have put 15 here. That would make it if I put 15, then again, it's still 5, right? So each of these, when I look at the rate, it's a constant 5 milliliters per minute. Where over here, it's 10 milliliters per minute. Except for this last one, which I again messed up. Over here, it's 10 milliliters per minute. Well, how did I transform that data? I just divided milliliters by minute for each one, and I noticed that the rate at which, no matter how many minutes I go on, every minute I'm producing 10 milliliters. So the rate, it's 10 milliliters here, and the rate is 5 milliliters per minute here. Now, why is that a suitable transformation? Because I'm able to see how much CO2 is being produced at a given time. Why is this transformation then, why would I think this transformation is appropriate? Because it's going to allow me to compare how fast these seeds are making CO2 at different temperatures. I have no problem with you just going and writing down what I said here, if you hopefully understand some of it. I think this was possibly this particular section B of problem 2 may have been a little bit over your heads because of so many different components, but hopefully you were able to put this together. May produce. Here, I think it's pretty straightforward. Here, I think you can do it. I don't really think you need me to do much here. For percentages, what you're going to do is, with percentages, remember, you're taking whatever it is you have and divide it by the total. So 124 divided by 159 is 78%. Then, of course, to find this percent, you would take 159 or 35 and divide it by 159. That gets you this number. And, of course, the total, what percent is the total? 159 was 159 divided by 159. So it's 100%. And the same thing goes here, except you don't have the total. So how would you find the total? You'd sum it, 115 plus 26, and you'd go ahead. Now here, these totals don't make a lot of sense, except that maybe you want to add the total number of the two total numbers together to get the total here. That's the only thing I would say to do. So here you see 9 minus 8, and that's because you have your centimeters cubed, by the way, is the same as milliliters. So it's 9 milliliters, or 9 centimeters cubed, minus 8 centimeters cubed. So that gets you 1. Divided by 5. Why? Because it took 5 minutes to get that change. So 9 minus 8, that's 1, divided by 5 is 0.2. 0.2 what? 0.2 milliliters, or centimeters cubed per minute. There's that negative 1 again. Okay. All right. And the same thing goes here. If you have 8 minus 7.2, that's going to get you a number divided by, again, how much time passed? 5 again. And that's going to get you this number. And then 7.2 minus 6.2 divided by here. This was another 5 minutes passed. So again, it's 5 minutes. This was 1, so you can keep doing that math. Here, it's 1 divided by 15. Where this is photosynthetic rate. Okay, so, and this is average time the leaf disc was allowed to float in 15 minutes with a light intensity of 100. And this time was taken for the leaf to float. The reciprocal gives a per minute rate. The variable measured in is the time taken for an event to occur. So if you have a light intensity of 100, average time, reciprocal time is 0.06. Now, I guess the question is, that some of you might have, is why are you taking 1 and dividing it by 15 here? because 1 is 100%. So you take 100% and you change it to a decimal form, and that. So what's 100%? It's a 100 over a 100, and what is that equal to? Well, that's 1. What's 50%? It's 50 over 100. So that equals 1 over 2, or 0.5. Right? And so what's 25 over 100? That's equal to 0.25, etc. And 11 over 100 is 11%. And you can do the math on that. And every time you have those decimals, you can divide, 1 divided by 15, and that's going to give you a number. And you could take 0.5 and divide it by 25, and that'll give you a number, etc. The frequency and size of size classes in a sample of eels. All right, so you have a size class of 0 to 50. You see 7 of the eels in this class, so the relative frequency is 2.6%. Well, why? Because the total is 270. You see 7 of them, so you're finding what percent. How often do you see these? Well, if you see them 7 out of 270, that's 7 divided by 270, that times 100 to make it a percent, you have 2.6%. So each one of these you do the same. You take 23 here and divide it by 270, and you get some percent. That's the number times 100, and that gives you a percent. 59 divided by 270, because this is the total here. That's the total. And each of these then would give you a percent frequency for all of this in this chart. So it should be pretty straightforward. So here you have dependent, independent variables. You have preparation, execution, recording. These are just kind of simple background concepts for when you're doing any lab. So here you have a chemical experiment where you're using an enzyme. Here's an enzyme. You can do the reading as to what the enzyme does. It takes and breaks down materials. It breaks down hydrogen peroxide into water and oxygen gas. And so at the different temperatures, the rates at which they're produced are measured. All right. Well, what you have to ask yourself, well, what's happening? The entire experiment is described here. And again, the height of the bubbles, probably in millimeters, is going to be measured here. So obviously, this is, number one, at 10 degrees is not as high as it is at 20, and the highest seems to be at 30, but then at 60, it's not as high again. In fact, it's less than it was at 10 degrees. To write a suitable aim for this experiment. Why would you want to know? What are you trying to do with this experiment? Well, I hope you can see that you want to find the temperature at which this enzyme works the best. And we'll talk more about enzymes as we go along. The whole point here is not you understanding enzymes, but you understanding the design of the experiment. So I think you can see that you're measuring how fast, by measuring height, you're measuring how fast this reaction is happening, or how much, in some way, measuring how much this gas is being produced in a given amount of time, right? So you're looking at the rate of gas production at different temperatures. So you're trying to find out how well catalase works. So you don't know what an enzyme is, but you know the name of this thing is catalase. How well does it work in producing oxygen? So the next thing is write suitable hypothesis for this experiment. So you might write something like, if the temperature is increased then the catalase will improve its function. So that's kind of straightforward little if-then statement. This shouldn't be an issue. Now, of course, that particular number degrees at 60 degrees, this particular hypothesis doesn't explain 60 degrees. 60 degrees has to do with the denaturing of the enzyme, but that's something for another day. So let's go on and discuss then this part 2. So part 2 here, or question 2 says, write a suitable hypothesis for this. I wrote the hypothesis in the wrong spot. This should be here, and the aim of the experiment is to see what's the optimum temperature range for the functioning of catalase. That would be what should be for number 1. Identify the independent variable, the variable you're changing. Let's look at that. What is the variable you're changing? Is it the height of the... Are you changing the height of the bubbles? No, that's not what you're changing. What are you changing over and over again? I hope you said the temperature, because it is in fact the temperature that you're changing. So you're changing the temperature so that you makes that your independent variable. State the range of values for the independent variable. Well, the range of values is 10 to 60, right? 10 degrees Celsius to 60 degrees Celsius. Name the unit for the independent variable, degrees Celsius. List the experiment needed to set the independent variable and describe how it was used. Well, what kind of equipment would you need to do this? Well, obviously, hopefully you can see a test tube. You'd probably need a graduated cylinder. Why? Because a graduated cylinder is used to measure volume. You would need a test tube, obviously. You would need catalase, which is the enzyme. You would need hydrogen peroxide, some volume of it. You would need a cooler or a warmer to make it go from 10 to 60, some kind of warm bath. I hope that makes sense. Something's going to allow you to change the temperature, a test tube, and of course some kind of ruler to measure the height, or something, a grease pen to mark mark the line on the test tube. So list the equipment needed, and we just measured that. Well, to set the independent variable, you would need some kind of warm bath, because you need to set the temperature. You would need a thermometer or a temperature probe and some kind of warm bath to help create that temperature. So identify the dependent variable. Well, I hope that you can recognize that the dependent variable was the one that's changing because of the temperature. The catalase is staying the same. The hydrogen peroxide, the water, and oxygen are being produced. So what is changing because of the temperature change? Well, it's the height of the oxygen bubbles. List the equipment needed to measure the dependent variable. Well, a ruler, some kind of pen to mark it, and you're ready to go to mark it on the test tube. Each temperature represents a treatment, sample, trial, circle 1. Each temperature represents a treatment. What's a treatment? It's a condition in which you are putting the things you're measuring. The sample is the material that you're putting in there, and the trial is how many times you've run it. State the number of tubes in each temperature. There's 2 tubes, tubes 1 and 2, right? 2 tubes. State the sample size of each treatment. The sample size, I believe it said it was about, 0.5 milliliters of enzyme and 3 and 4, so 0.5 and 4 milliliters. Remember, milliliters are the same as centimeters cubed, right? Centimeters cubed is equal to milliliters. So 0.5 milliliters, 4 milliliters, so together 4.5 milliliters is what you have in your sample size. 0.5 milliliters of enzyme and 4 milliliters of hydrogen peroxide. State how many, state the sample size, so 4.5 milliliters. State how many times the whole investigation was repeated. Let's see. So the whole thing was done twice, 2 separate days. So the whole experiment was repeated twice. So they wanted the whole experiment, not just each trial or each sample. Explain why it would have been desirable to have included an extra tube containing no enzyme. Well, I hope by this point you can talk about a control group. Why would it be important? So that you can show that the bubbles don't appear when there's no enzyme. So that's key. That shows you that the catalase is what's doing is what's allowing the bubbles to form. It's your control. So the no enzyme would be your control group. Identify 3 variables that might have been controlled in this experiment and how they could have been monitored. All right. So you're changing the temperature. So perhaps the amount of light present, the volume used. Light would be obviously conducting the experiments in the same conditions or same area. The volume, careful measuring. What else would you want to try to control? How you add the enzyme to the hydrogen peroxide. The method of adding. You don't want to just dump it. Perhaps you just pour it carefully. The technique of pouring, you want to keep it the same for each. Why would it be important to control light? Because maybe light has an impact on hydrogen peroxide turning into oxygen. It might break down hydrogen peroxide without the use of catalase. The volumes used should be the same in each one, because if you add more catalase or less catalase, it could impact the bubbles, and you wouldn't know if it's the temperature or if it's the amount of catalase. So you have to be very careful with that. And how you pour it, again, the violent pouring versus a easy pour might make a difference between getting a lot of bubbles, or you could create bubbles just by pouring it. All right. So that could confound your results, right? Let's go to the next one. So here is an experiment that was done. They increased the amount of fertilizer that they're adding, and then this is the sample size. So they got mass of the radish. So these are, and of course they're looking here, and so in sample 1, with no fertilizer, you got 80, a mass of radish root of 80. In the second sample, you had a mass of 83, so around 80 for all of them. You had 0.06 amount of fertilizer, and you get this mass, right? And you had as much as 0.3, and all of a sudden you get these masses here. It's interesting to note that the mass actually went down as you went from 0.24 to 0.3 grams per liter. Again, there's that L with a -1, meaning grams divided by liter. That's the concentration of fertilizer. So you have a total mass of 408 and a mean or average mass of 81.7. So here we're manipulating, or we're transforming the raw data. The raw data are these masses that you're measuring, where here the total mass is summing, and here the average, the mean means average. The average mass for each of the different samples is listed in 2, right? So these are 2 different data transformations. So I think you can calculate the total mass and the average mass for all of these. How do you calculate averages? You take the sum, in this case 408, and divide by the total number of times you've added them, right? So you added 5 different radish samples, you got 408, so take 408.5 divided by 5, and then you're going to get 81.7. So take your total and divide it by 5, and that's what you're going to get here for your mean, or average mass. All right, then please read each of these so you understand what's going on. Well, the independent variable, I hope you know, is the fertilizer concentration. Right? And what's the range? Hopefully you know it's from 0 to 0.3 grams per liter. One of the radishes recorded in Table 1 did not grow as expected and produced an extreme value. Record the outlying value here and decide whether you should include it in future calculations. Well, here the extreme value is 23.6. You know, it's obviously grew way below what would be expected. I mean, should you include it in future calculations? You probably shouldn't. And the reason why we call it an outlier, right? So it's something happened to that. Maybe there was something wrong with the seed. Maybe there was something wrong with that. You know, maybe there was an insect that was eating it. Who knows what exactly happened with that? But obviously something went wrong. They had nothing to do with the fertilizer, because you can look at all the rest of the data and see that this number did not fit. You can graph it, but you can call it an outlier and just leave it out of your calculations when you're looking at your symbols, or at your transformations for your data. So complete the table on the previous page by calculating total mass and mean. You can do that. Use the grid below to draw a line graph of the experimental results. Use your calculated means, and remember to include a title and correctly label the axes. So your independent variable, the variable that you're controlling, all you're going to put on the x. So you always put your independent variable on the x. Well, your independent variable is, in fact, your concentration of fertilizer. And it's in grams per liter. Your concentration of fertilizer has to end up at 0.3, but I would suggest that you leave your last one should be 0.5 or even 0.6. But I'll say 0.5. And why 0.5 instead of 0.3, since your last data point is at 0.3, so that you can predict into the future what would happen if you see, if you continued on to 0.5. So if you have 1, 2, 3, 4, 5, 6, 7, 8, 9 different divisions, how many would each big line be if 9 of them is 0.5? Well, each one of these large lines would be 0.06 more or less. Suppose that's 0.06, so this is going to be. So if we set this at 0.5 being the max, again grams per liter, then we can go ahead and divide 0.5 by 9, and each one of these larger divisions is 0.056. And because each of these small lines is divided by is worth 10, then we can go ahead and divide that. You can just move the decimal one time to the left to find out what each little division is. Now, had we had just one more grid block here, so we had 10 by 10, then our lives would be a lot easier, because it would be 0.5, and then down one would be 0.4, down one would be 0.3, etc. All right, so that gives us our 0 to 0.5. Even though 0.3 is our largest value, we'll be able to extend the line and see what would happen if we just kept increasing the concentration of the fertilizer. The concentration of fertilizer at grams per liter. And then what's the average size? What's the range for the average size of the radish? So you would put the mean mass. You do the same thing that I did down here with concentration. You're going to do it over here with the mean mass. Right? So this would be the mean mass. I believe it's in grams. And so there, what's going to be your lowest value and what's going to be your highest value? You're going to have to figure that out after you've calculated the mean mass. Remember, mean just means average. So how do you calculate the average mass or mean mass of radish in grams? You're going to take the total that you've added up, the sum of all the masses with that concentration of fertilizer, and then you're going to divide by 5, because you have 5 different masses that you're taking the average of. So it's 81.7 at 0. So here's 0. So it would be 81.7. Where you would put it would depend on your range. So you have to see what your biggest value is. Your highest number is going to be about 150. It's probably the best number to set it to. There is nothing that's 150, but if you set it, number one, 150 is a nice round number. Secondly, it's high enough that if there's any values, if you want to extend that line to look at the trend, if it turns out that value is much higher, if it varies when you draw the trend line, you want to give yourself enough space where you can extend that line and still be able to get some numbers out of it. So the lowest number is 81.7. So you can safely put 81 as your lowest. But you might want to go ahead and say 80. Again, it just makes your math a little easier when you're starting to do your divisions. So you have 80 is your lowest and 150 is your highest. So there's 1 here, 2. I'm sorry, that's not 2. It's 1, 2, 3, 4, 5, and 6. So the difference between 80, which is your lowest value, and 150 is 70. So you have 70, and you're dividing it by 6. So the best thing you can do is just say that each major line is going to be about 12. It's 11.67, but if you just round it off to 12, it makes your life easier. So 80 plus 12 is 92. 92 plus 12 is 104. 104 plus 12 is 116. 116 plus 12 is 128. 128 plus 12 is 140. And 140 plus 12 is 152. So yeah, you're above 150, which was the goal, but the key is that at 152, you still have your range. Your data fits within that range between 180 and 152, and now you know, since there's 12 in between each major division, you know that there's about 1.2 for every small line, because this is, again, it's a 10 by 10 square, so each division is, each little line is 0.1, so it's 80. So it would be, instead of 12, it's 1.2, so it's 81.2 is here, 82.4 is there, etc., all the way to 92. So it really works out well, as long as you remember that you have to kind of go with the flow and do some rounding and decide how much are each major divisions are going to be worth to make sure that your data fits in this space. So I know my highest data here is 0.3. So my highest value on the x-axis is going to be around here. My highest value for the y-axis is going to be around here. So actually, if you think about it, it might seem like a lot of this is not going to be used, but if you extend the line past 0.3, you're going to use it. You're going to use this space. You're just not going to use it to plot your data. You're just going to extend that trend line so that you can tell what's going to happen in the future if you increase the value, the concentration of fertilizer to 0.5 or 0.392, etc. So in any case, you know that you have 80 at 0. So your first point is, I'm going to put the points in blue. Your first point is here, and you would then go ahead and find your concentration on the y-axis, on the x-axis, and then put your mean mass here, and you should get something. So you kind of expect the line to be somewhere along there, something that looks like this, and your data ends about here somewhere at 0.3. But you could then extend the line, as I said earlier, extending the line beyond the values that you've measured, and see that at 0.5 you can end up with less root growth. I mean, that's one possibility, of course. It's hard to tell. You might actually get something like this, kind of meandering around some plateau. You really would probably want to collect more data, but this might be actually too much. You might decide in your case not to use 0.5. Maybe you can set this at 0.3 and just say at 0.3 is the max, and then just say that each of these smaller divisions, if this is 0.3, then each of your smaller divisions is 0.4, 0.04. So that's 0.04. If you did that, then things would change a little bit. Let's go ahead and do that and see what it would change. So you saw there that if we set it at 0.5, we'd have that room to play around with and predict what's going to happen after our collected values are placed on the graph. But if we go ahead and get rid of these and say what happens if instead of 0.5 I set these at this value at 0.3. Well, once you set the divisions to 0.04, then what you end up with, and the reason it's 0.04 and the reason this is not at 0.3 is because 0.3 divided by 9 is a number that's not as easily manageable as 0.04. So by setting it to 0.04, I'm able just to add 0.04 for every big number, every big line division. Every smaller line is worth 0.004. Why? Because these are this grid, this graph is divided into 10 by 10, so 10 small lines for every large line, or 10 lines per centimeter. Alright, so there's your data, and of course you would start graphing it again. Looking at your line again, I'm just kind of sketching out a line here. When you're looking at your line, it's going to look something like that, very much like the one we looked at earlier. And if you wanted to extend your line, you could extend it. And so here you filled up pretty much your whole graph, and you're able to then maybe calculate a slope to see how much what's the rate of mass growth per fertilizer concentration. What's the mean mass of vegetable you get per gram per liter? So it would be grams of mass of vegetable for the concentration of fertilizer. How fast would you get? How much do you get for every so much of fertilizer? How much vegetable do you get? You also see that there's a decrease here, so that after a certain concentration, something happens that you actually get less mass. You might want to extend that line, do some more experiments with higher concentrations to see if that trend continues. So it says, describe some other measurements the students could have taken to make their experiment more complete. So they measured mean mass, they measured concentration of fertilizer. What else could they have done? Could they have measured time? Could they have measured the size of the plant? Root distribution, they could have looked at how far away each of the radishes were from each other. There's so many other things you could think about that I really... Maybe when we get together, we could come up with some different explanations. Here, to calculate the mean, median, and mode for each of these, we could do this in class together. But the mean is just taking your number of leaves for a concentration, adding them up. That's a sum, and then divided by 5. That's going to get you a mean. Calculating the median, you can put these numbers into a calculator and just say, give me the median, or into Excel. We'll do that in class. I'll show you how to do it. So if you put this grid into Excel, you can actually calculate the mean, median, and mode just by putting in the formulas. Which concentration of fertilizer appeared to produce the best growth results? Excuse me, it's getting late. Again, I think we could finish discussing this in class, but You have to look at the data, look at the mean number of leaves, look at the median, look at the mode, and use that information to really think about how, how well these plants grew in these different concentrations. Describe some sources of error for the experiment. We have an outlier. We have one that did not grow. What could be some errors that you might run into? Perhaps perhaps some of these plants could naturally have better genetics than others. That could influence how fast they grow with a given fertilizer. Measuring errors in how much fertilizer you're actually measuring the concentration of fertilizer. Counting the number of leaves, maybe people miscounted. So those are sources of error. Write a brief conclusion for the experiment. Include a reference to the aim of the results. So your conclusion, after you've looked at the data, you might say something like, an increase in fertilizer increases the mass of the plant up to a point after which the mass seems to decrease. The mass of the root seems to decrease. And actually calculating mean and mode. Again, you can look up here, median, mode, and range, and think about what those numbers mean. And remember these, all right? Remember these rules as far as calculating averages. Or means, I should say. All right. A brief conclusion for the experiment include a reference to the aim of the results. And what's the aim is to see what's the best concentration of fertilizer. And I think you can write your own conclusion. The students decide to replicate the experiment, carry it out again. How might this improve the experiment's results? So they're going to do it again. What might improve? Well, you might get rid of that outlier. You might be able to get all of them germinating. You get 100% germination. You get more accuracy. You're going to get more accurate results. You're going to get closer to truth. The more results, the more times you repeat the experiment, the less any one error is going to impact it. Remember that one outlier, that one that was really small for whatever reason, and this one that did not grow, they impacted the results if we allowed them to calculate. Even though we didn't include this one in the calculation. I forget what concentration it was, but that one that was very small, we left it out of the concentration, the calculations. Even though we left it out, remember now instead of dividing by 5, we're only going to divide by 3, so our mean is going to be smaller, or by 4. So our mean is going to be impacted by that. So if we do it again and we add 5 more, so now instead of dividing by 5, we're dividing by 10, the impact of any one error becomes smaller and smaller the more we repeat the results. So the mean gets closer and closer to the truth, or we get more and more accurate results. Okay, that's it. It's a long time coming, but I'm finally done. Went over all of these. We'll go over the highlights in class. If you have any questions, feel free to send them to me. All right, have a good one. As we move forward, as you look here, it says the more you turn the knob, the larger the flame, the bigger the increase in temperature. The independent variable, which is whatever, is the variable that you change. It is not dependent on the other one changing. It's dependent on you change. You change the independent variable, whatever that is. It's not dependent on the other one. Does that make sense? So in the flame example, the knob is the independent variable. Turning the knob is the independent variable. So that's an example that I think is clear and simple. So now let's look at the clothespin. Another example. Let's look at the clothespin. In the clothespin example for the bag, the test would be something that changes when metal is involved but does not change when metal is not present. So we need to think about what are the properties of metals. So we have conducts heat, conducts electricity, malleable. Malleable means you can bend it. That's why it makes good, metal makes good jewelry. And magnets are magnets are attracted to many types of metal. So for this example, and for this example only, I used the magnet test. So I said the magnet must test to see if there's something in the bag. If there's a bag here that has tape around it, and it's numbered, and I'm going to test if there's metal by moving a magnet toward or away from the bag. So change the distance from the bag to the magnet. It's going to be connected to some kind of force meter. And the meter is going to measure this in newtons. That's what I'm going to do. That's my setup. That's my experiment. So in this magnet test, I'm going to measure different distances. So I'm going to have 10 centimeters, 5 centimeters, 2.5, 1.25, 0.625, 0.3125. So I'm going to change the distance, and I'm going to see how strongly is the magnet getting pulled towards the bag. If there's no metal in the bag, what's going to be the force? Zero. So if there's no metal in the bag, then all of these would be zero. If there is metal in the bag, then it's going to change. Probably as I get closer, the stronger the pull, right? So maybe I start off at 0.1 newtons, then 0.5, then 1.5, then 2, then 2.5, then 3. So these numbers, you're not actually expected to calculate them. I'm just giving you examples. So when you're looking at these numbers, it's just an example. Okay? This is just an example table that describes what's going on in the experiment. This whole experiment's just an example. But you see, I did this. I created this based on just simply looking at the bag. That's all I did. And you could have done the same thing. Is that correct? Yeah. That wasn't that hard. All right. So the next step is answering the rest of this. It says we could repeat this and collect data several times, and we would, if we were actually doing it, we would repeat this several times. Then we're changing the distance from the bag. We're changing the different distances. So the distance is what? What is the distance? Is it dependent, independent, or control group? It's the independent is correct. The distance is independent. Why is it independent? In this experiment, who changed the distance? Did the distance change because of the force? No. We changed it. So because we changed it, it's independent. Yes, Perry? You may go. Sign out. Make sure you wash your hands before and after, and return the password goes, please. Thank you. According to this hypothesis, the object, and I meant to put this, is, not tis, is a clothespin, then there is metal, right? We're assuming there's an assumption there. You agree? If there is metal and it's magnetic, then the magnet gets closer. The closer the magnet, the stronger the force. Everybody agrees with that, right? So, what is the force? What do we call the force? What kind of variable?
Dependent.
The dependent. Why do we say dependent? Because it's changing as the distance changes, right? We change the distance, but then the force changes. So because of the distance, and therefore it's the dependence. In other words, the force is dependent on the distance. All right, now we would go and keep the other variables the same, and because we're keeping all the other the same, what do we call these?
Control.
Control or constant. I like constant. Do you agree? Constant variables. But I also, another word for it is controlled variables. And unfortunately, both words are used, so you should know what they are. You'll see this kind of stuff on the ACT, SAT, end of course exams. You're going to see it on all kinds of tests. And by the way, this stuff comes up in psychology and in the sciences. It even comes up in business school. Bless you. In law school. No matter where you go, this stuff comes up. What does the bag represent? That's the question.
Was it how Einstein taught his class?
That's the history of it.
It was like a shield to cover with.
Yeah. I hear your answers. Go on. When I hear one that's
It represents like science is not knowing like exactly what it's
exactly. It represents the unknowable.
That's awesome.
That's right. The fact that we can't see things, we can't be 100% certain about stuff. That's what we're saying. That we can't be 100% certainty. There is always uncertainty. Did I show you guys the elephant and the 6 blind guys?
Yeah.
All right. So it is literally just like the elephant with the 6 blind men trying to figure out, feeling different parts of the elephant, everyone being 100% certain they were feeling a wall or a rope or a snake or a tree trunk or a leaf, when actually they were feeling an elephant. And the point is of the story, and the point of this, one of the main themes of this lab is that it's very difficult, if ever, to have 100% certainty. So for those people that haven't written a hypothesis, if you didn't get a chance to manipulate the paper bag, I need you to take a paper bag home with you. I need you to promise not to open it. I'm going to go ahead and take you at your word. It really doesn't matter if you open it or not, people. It doesn't matter. You realize that, right? But I'm going to put you on your honor and don't open it because it kind of ruins the experience for yourself. But the whole point of the bag is, based on your observations, you should be able to formulate a hypothesis, if-then statement. Think of something. Well, I gave you an example. You know, I'm not going to fill this in because this is going to be different for everybody. This part's going to be different for everybody. Does everybody get that? So this is going to be different for everybody. All you got to do to get this right is for it to be logical. Logical. That it makes sense. Right? It's one part is dependent on the other and makes sense. So what's a good example? Well, what I told you here. I gave you an example right here, and I said, if the object is a clothespin, and why did I pick clothespin? Because people were saying it's a clothespin, so I said, okay, let's pick a clothespin. Then there is a metal spring, right? Because that's how a clothespin works. Do you agree? Mm-hmm. And because there's a clothespin uses, and see the because, do you see that? There's because, there's the if, there's the then. Because that is what a clothespin uses to keep the two parts of the clothespin together, right? That's how you pin things. You need a spring. And it's metal usually. Now, are there clothespins that don't have metal? I don't know. I don't care. That's not the point, all right? The point is that let's assume that we're assuming that clothespins require a metal spring. Let's assume that. If that's true, if there's a clothespin in that bag, then it has to have metal in the bag. Do you agree? Yeah. Okay, so if there's a clothespin, then there's metal in the bag. That is the hypothesis. What about what?
What about the Jenga block? There's nothing that really has besides wood.
That's what I said.
Right. And you want
How about
to the
what?
What about what?
The Jenga block.
Magnet is not.
Like there was like
The what block?
a solid piece of like wood. It's
There's a solid piece of wood in there, and so you have to come up with a way that you're gonna first come up with your hypothesis. You think it's a solid piece of wood.
Yeah.
So don't see. You see that's another thing that we're kind of trying. I'm trying to get to your.
But how would you test it?
So there's a difference between an observation, right? Because no one here observed the wooden piece of block, unless they opened the bag, right? No one observed that. There's an observation. Your observation is a hard. It's hard and square. You can feel that. So because you can feel it, you can say that. Right? Cellular or rectangular, whatever it is. That's your observation. Your inference, an inference is a guess. Your inference is a wooden block. So every time somebody tells me that, what about the wooden block? It drives me crazy. You don't know it's a wooden block.
Okay,
all right.
If there is one, how would you test that?
A good question. It's hard to test that, isn't it? Because it's not as easy as metal. That's why I picked what? What did I pick? A metal. Because metal's easier to.
Can we use the same one as you?
So every bag
I thought it was the same. Because my hypothesis has a cluster, but it's not the same.
All right. So I heard a good one for wooden block. If you want to hear me.
Yep.
All right. So they thought about it and they said, well, what are some of the characteristics of a wooden block? If it is, and an inference is what you want here. You want an inference there. That your hypothesis is your inference. So you think your observation was as hard and square and it's a wooden block. So what are the qualities? The question is qualities of wood.
There's a unique sound when you hit it to get something.
Okay, so there's a sound. A unique sound, so that's good. Okay, it's kind of an almost hollow sound, but not quite hollow. Okay, so it's a unique sound. What else? Its density is
Hard.
well, density is it's not going to be hard or soft. Its density is going to allows it most of it to float. Do you agree? Wood floats?
No.
Wood doesn't float?
It does.
It depends on how.
What do we make boats out of?
But that's
No problem. It's okay. It's okay. Hey, you guys gotta get comfortable with being wrong. All right? It's okay. I'm gonna be wrong. Yes, it's okay to be embarrassed about being wrong. But being wrong is part of learning. If you're not wrong, you're not learning. So it's okay that you're wrong. It's okay that you fail in your practice. You fail in your practice so you don't fail in the test. Okay? You guys have to start to learn to be comfortable with that. All right. So sound is unique. Density allows it to float. What else do we know about wood? Does not break easily. Yeah.
It's 11:57.
Are you serious?
Yes.
Oh well. And this is why I don't like spending a lot of time. Guys, honestly, can I give you my honest opinion? If you guys would have went online and looked up what I sent you, the step-by-step how to do all the stuff I taught you to do, you could have done it on your own. Do you agree?
Yes.
Okay. All right, so we'll continue this tomorrow. Obviously, listen, before you go, obviously a scientific method packet's going to have to be due next week, not tomorrow.
Thank God.
But we're going to go over this, okay? But you guys, if we're not getting efficient, it's not going to work. We have to be efficient. I'm sorry, say that again.
Can you list the rest of the questions that we studied for the test?
One question from each page of the syllabus. You pick. You pick the most important stuff. I'm going to test you. You might study the wrong stuff, but that's your problem. I'm not sure, Perry. What can I say to you, Perry?
Yes.
So the changes as you change one variable, a dependent variable is a thing that changes as you change one variable, it changes, right? So it changes as you change one of the variables. So let's say that you turn the knob on a stovetop to a higher setting, allowing more natural gas to combust and the flame in the stovetop grows larger, the dependent variable would be the temperature, right? So as you turn up the knob, that's turning up the flame, right? Turning the knob turns up the flame. What increases because you turn that knob? The temperature, right? So the temperature is dependent. Depending on what? On how high you turn the knob. Does that make sense? So in this case, the temperature is indeed the dependent variable. So now as we move forward, let's go into the next slide then.
Are they red or are they green?
Okay. So still, every science experiment is a group that we can use as a control group. You can read this yourself. I don't want to go into it. I think I wrote it pretty well, but whatever. We can then create a duplicate bag. So the idea would be we create a duplicate bag that we know has the control, that has the clothespin in it, right? We know there's a clothespin in this bag, and that clothespin has the metal. All right, so we know it has metal. There's tape around it, so it looks exactly like the bag that we don't know. Okay? And we're going to move the magnet closer and closer to the bag. And so that allows us, we know, this is called what kind of control is that?
Positive.
The positive control is correct. So this is the positive control group. Why is it positive? Let's see, clothespin. One, the control group, putting a clothespin. Putting a clothespin in. All right, so that has it has the control, so it's positive for the pin. What about this one? We can do the same thing, but this one we make sure there's no metal in it.
Negative.
So this is a negative control group. It's negative because there's no control group in it. There's no metal in it. So what? How would we use that? Why do we even bother doing that? We bother doing that because by the end, we're going to have three groups. We're going to have one group, and let's assume that they're the same. And unfortunately, it's not. It doesn't look like it, but it is. So they look exactly the same, as exactly as we can make it. And one is an unknown. We don't know what's in it. One has the clothespin, the other one doesn't, and we use the magnet setup just for all three of them. And if this one is attracted to the bag, this one's not attracted to the bag, and this one is attracted to the bag. Well, this is the positive control. You're right that it's positive. So this one, group A, is showing us what it's like if there was metal in there, if there was a clothespin in there. And this group C is the unknown. We don't know what's in it. And group B is the negative control. This is how it should act if there was no metal in it. And so what we can tell based on this experiment is not that there's a clothespin in there, but that this bag behaves as just like this bag behaves. And so because of that, we can say that there's evidence that suggests that we have metal in the bag and probably a clothespin. Probably. Not definitely. Is that clear? It's unknown. We can't ever know. So the rest of this, you're going to go ahead and answer this if you haven't. Now let's spend some time going over then the scientific method.